Well, I'm just gonna hide this.
How are you? Good. Guys gonna eat that one?
Yeah, I'll wait. Are you?
Thank you. So probably have to use my laptop.
So just.
Yeah, on the person with the blood. Thank you.
And then I'm mainly just maybe stay here.
I mean, if you want to use it. Yeah.
Sometimes I like to move, but I'm loud enough, like it's.
Hey, how are you? We're gonna meet right after.
A little bit.
Oh, yeah, I gotta talk, it'll be great. Okay, good to see you.
Yes, you're back. I think it might be easy to use this. People in the back. You're gonna be like, listen to it. I mean, if you want to move it. That's all right.
So I would turn on the presentation.
Yeah. But, I mean, the speakers, you can speak into this. It's just that if you're moving away from this here, this one goes to the Zoom.
Okay.
Yeah. Thank you. Lita should be coming down and introduce herself.
Just takes a lot of air. Yeah, very few.
Sorry. Ask yourself.
Oh, you're just.
Oh, here.
I know. Everything.
You can get it on top.
The minute of.
I don't want to.
Oh, it's.
No, it's good.
It's like.
Yeah, Lita should come down and.
Let's like the Canadian. It's just well, it's like this.
Thanks for it.
Yeah.
You plans for the years?
No. The paper is right here. And then, oh yeah, you can. There's people in the room already, but I think more people are like slowly filling it if you want to. And then you can speak it. This one, please.
Like how can we? Hi, everyone. Let's start in about 1 to 2 minutes.
Yeah, absolutely. Some of my slides before something.
All right, thank you, everyone. Where do I speak here? Thank you, everyone, for joining us in this academic year P30 seminars. We are really excited to start this year with Donghai Liang. Most of you may know him, or you may have already collaborating with him. I mean, he's Associate Professor of Environmental Health at Emory University. He's an exposure scientist and a molecular epidemiologist. He led several NIH-funded grants in his short tenure, and he published more than 120 articles. I mean, these are just, you know, numbers. The reality is that Donghai is a leader in environmental omics and exposomics. He worked really hard to come up with some cool ideas that you're gonna hear about on integrative multi-omics. And why do we need this to uncover, you know, the biological mechanism linking environmental exposures and outcomes in several different populations, vulnerable populations, and different life stages. So I hope you will enjoy it.
Thank you so much, Lita, for this really nice introduction, and thank you so much for everyone to attending this seminar in person and online. My name is Donghai Liang. I'm very happy to be here, actually visiting USC for my very first time. I'm super excited. And as Lida mentioned, I've been working with many of the great faculty members here. I see so many great faces, and I'm excited. So I'm currently directing the environmental metabolomics and exposomics research group. I call it Emerge at Emory. We are a group of passionate molecular epidemiologists and exposure scientists trying to use both targeted and untargeted exposomics profiling to characterize the individual exposure across the lifespan and trying to use single and multi-omics technology to understanding the different environmental exposure listed here and see how they affect maternal and child health and vulnerable population with chronic diseases such as Alzheimer's diseases and cancer. The motivation for us to really conduct this research lies in the serious public health burden caused by different environmental exposures. So in traditional environmental epidemiology and environmental health research, we tend to focus on single group exposure and single health outcome. However, this is not enough when we think about intervention, when we think about how to solve the real problem to protect those highly exposed populations. So a knowledge gap really lies in the question mark here, which is the underlying mechanism. So when we are exposed to higher level of exposure, how can we develop or how come people are in higher risk of developing these devastating diseases? So to truly understand and intervene, in my opinion, we really need to move beyond the association and also couple with systematic biology and omics-driven approach that can uncover the molecular pathways and mediators that bring the exposure to the health effects. So this particular challenge is even more difficult to solve when we enter in the era of exposome. So you might have heard about Dr. Lida Chassi and Dr. Rima Haller talking about exposome all the time. So this is really the concept of like characterizing the totality of exposure each individual can experience this across the lifetime. So everything from the air we breathe in, from the water we drink, from the food that we ate, and from our social neighboring environment, these are all affecting our different risks of developing diseases. And this exposure, oftentimes they do not act alone. They interact with our biology in complex and dynamic ways, and you will be hearing me talking about different omics, so they affected our genome, epigenome, transcriptome, proteome, metabolome, lipidome, and microbiome, along with other complex internal systems. And these are just a small subset of adverse diseases that will come through this exposome's impact. So this big challenge we face nowadays in environmental health research or even exposome field is how to link this vast diversity of exposure, understanding their role in triggering all these diseases. And that's what motivates us to really try to use this omics technology in this integrated exposomics field to find out the biological underpinning puzzles or roadmap that bridge and connect the exposure to the disease outcome. And thanks to the development of high-throughput omics sequencing and analytical technology, many omics-based approach can potentially tackle this challenge, and they are shown here nowadays. So for my talk today, I will tell you how we apply each of them, hopefully, and then I will start with metabolomics as an example. So what is metabolomics? Metabolomics is basically the large-scale global detection of small molecules commonly known as metabolites in our body. The reason why I started working with metabolomics 11 years ago is because if you look at the human metabolome, you realize that this is really a downstream product of the gene transcription and also the gene-environment interactions. And therefore, the biological changes in the metabolome are oftentimes amplified after all this environmental exposure interact with the genes. And also as downstream product, the metabolome is also closest as a functional readout of all these health responses that we oftentimes observe. And 10 years ago, I started working really closely with Dr. Dean Jones and his lab at the clinical biomarker laboratory at Emory, where we developed an efficient, high-resolution and targeted metabolomics workflow that's capable of detecting and characterizing tens of thousands of these small molecules in less than 125 microliter of samples. So together, our team developed several different biostatistic tools, including in the feature extraction, in the metabolome association study, in the pathway enrichment analysis, and in the chemical annotation phases. And I'm going to tell you a little bit more about that later. And using this powerful platform, my team has successfully applied environmental metabolomics in more than 20 different independent cohorts. I can keep talking on, and maybe you won't be able to enjoy your weekend. So I will spare you with that. Today, I'm going to tell you just a few studies to highlight some of our work. So I want to first focus on maternal and child health. The reason is that, as we all know, pregnant mothers and newborns are oftentimes the most vulnerable to these different environmental impact during the pregnancy and during the early life. And then ranging from air pollution to persistent organic pollutants, we're seeing so many adverse health effects coming out from exposure to these environmental chemicals. However, this health burden is oftentimes not shared equally across the population. As you all know, like people of color, such as Black and African Americans, Hispanic population that constantly exposed to disproportionately higher level of environmental pollution and also experience the highest rates of adverse pregnancy and birth outcomes. However, there's limited information on the biological mechanisms underlying this health disparity due to the lack of sensitive biomarkers, lack of comprehensive design. So to address these knowledge gaps, really thanks to our cohort PI, Dr. N. Dunlop, and our team at Emory, we received multiple NIH funding over the years to conduct this exposome and multi-omics study in the Atlanta African maternal and child cohort, trying to solve some of this puzzle that where is really the underlying mechanisms connecting environmental exposure to adverse birth outcomes in this minority population. And let me tell you a little bit more about this cohort. So the Atlanta African American maternal and child cohort, I would call it ATLA cohort from now on, is an ongoing prospective cohort that started in 2014, and we are still recruiting people. There's more than 700 pregnant mothers and their newborns diets that are recruited so far. And as you can see here, this is a socioeconomically diverse population, and it's also a highly phenotyped birth cohort. As you can see, we have almost 20% of the mother deliver small or gestation age baby. Another almost 40% of the mother deliver either preterm birth or early term birth baby. Within this cohort, thanks to multiple funding support, we are able to complete both targeted and targeted exposome analysis from the geocoding, where you look at conducted this external exposome profiling, looking at air pollution, green space, climate change, heat exposure impact on pregnancy. We also are able to conduct targeted quantification on different environmental chemicals, including PFAS, phthalates, smoking, marijuana biomarker, environmental phenols, PAH, pesticide, and PBDE. In addition to the exposome profiling, we are able to conduct longitudinal biomonitoring on collecting maternal sample from early and late pregnancy, dry blood spot from the newborn baby within 48-hour delivery, and longitudinal biomonitoring among the children, where we conducted multi-omics profiling, including DNA methylation, metabolomics, lipidomics, microbiome, and so forth on the mothers, and then metabolomics and microbiome on the children. Then as for the clinical outcomes, we really are interested in characterizing birth outcomes, pregnancy complications, looking at how this newborn characteristic and the demographic factors and nutrition kind of play a role in modifying this environmental exposure to birth outcomes complications. We also focus on infant neurodevelopment and childhood obesity. And within this cohort, I was very lucky to get the support from the cohort. And 5 years ago, I got my first NIH extramural funding. This is coming from the ECHO Fund, where I conducted and decided this PFAS exposure and child health study. It literally took me a week to come up with this acronym, but I'm very proud of it. The reason is it's a peach study. I'm not sure if you guys know about Atlanta. It's in the peach state of Georgia. And then for this particular study, we focus on PFAS. You have know about all this amazing work conducted by Dr. Chazi, Dr. County, and Dr. Goodrich, and Dr. Chen on PFAS effects. We know that it's a group of long-lasting chemicals that are really hard to break down in the environment. And numerous studies have revealed that exposure to PFAS during pregnancy will lead to different adverse health effects, including negative pregnancy and birth outcomes. And our hypothesis is that this elevated level to PFAS during the pregnancy will result in some perturbation in the maternal metabolome, which will then in turn increase the mother's risk of delivering a baby that has some sort of fetal growth restriction. So that's the hypothesis of this pitch study, and we're going to use like targeted metabolomics trying to understanding the underlying molecular roadmap. So 3 years ago, one of my very first PhD graduate trainee, Dr. CJ Chang, she published this very first study looking at the metabolomics link between PFAS and fetal growth using a meet-in-the-middle approach. So here, she focused on the PFAS and a high-resolution metabolomics data collected during the early pregnancy. And then she followed a meet-in-the-middle approach by basically running 2 independent metabolome-wide association study, with one focusing on the PFAS level during pregnancy and the other on the fetal growth, and where we used gestational age, small for gestational age and low and the continuous birth weight as a surrogate to look at the fetal growth restriction. Then we were basically on the pathway enrichment level and on the chemical annotation level, trying to comparing the overlapping pathway and the metabolite associated with both PFAS exposure and the fetal growth outcomes. And let me tell you a little bit about the overall environmental epidemiological association. In this study, we observed that there's a significant association between PFAS levels and small for gestation age and low birth weight. Specifically, we found out that with the increase in PFOA and PFNA, 2 of them very common PFAS congeners, we are seeing that with the increases in this PFAS level during pregnancy, we are seeing a significant decrease in the low birth weight, and consistently, we're also seeing a significant increase in the odds of small for gestational age. Now the question becomes whether or not we can use metabolomics to detect any pathways or metabolites that could be explaining how this is happening. So using metabolome association study, we conducted pathway enrichment analysis that were on hundreds of metabolic features that we found out to be associated with both PFAS and gestational age, small gestation age. And one thing that we noticed is that there are 20 metabolic pathways that seem to be associated with both exposure and the adverse birth outcomes. And more importantly, this underlying pathway seems to be centering around dysregulation in amino acids metabolism, in energy metabolism, in bile acid, fatty acid, and sex hormone metabolism. And when we're trying to identify this metabolite, enrich this pathway, we compare them with the chemical standard, and we are able to confirm the identity of 10 metabolites shown here. I apologize, the text might be too small to read, but the take-home message is that these 10 metabolites seem to be associated with both PFAS and small for gestation age or low birth weight. I'm going to use uric acid as an example. So this uric acid is a pro-inflammatory factors. What we are seeing is that with PFOA and PFNA increases during the pregnancy, the intensity of uric acid also increases. Simultaneously, once the uric acid intensity increases, we are seeing that it's in association with a strong association with a baby born with low birth weight, and a marginally significant association with increased odds of small for gestational age, in which, again, this molecular epidemiological association is inconsistent with our overall environmental epi association. Not only do we see this pattern for uric acid, we also observe other potential metabolites that could serve as potential intermediators with linking PFAS to fetal growth, such as glycine and taurine in the amino acid metabolism, unsaturated fatty acids, some of the bile acids, as well as the anthogenic hormones. So which are all of these are closely involved in oxidative stress, inflammation, and placenta transport function. And this dysregulation might explain why these pregnant mothers were exposed to increased level of PFAS while on earth they deliver a baby that seems to have like a small gestation age. But again, as much as we are so excited about these findings, we need to keep in mind that this is a hypothesis generation study. So we need to replicate them. So one thing that we think about to do some internal validation is to conduct a follow-up study. This is led by Dr. Caitlin Tai Taibo, who recently graduated from a program and became a EIS officer at CDC. So what she do here is that 2 years ago, she led this study. Instead of looking at the maternal metabolome, she focused on the newborn metabolome, looking at the dry blood spot collected from the baby within 48 hours of delivery to see if PFAS exposure during pregnancy would do anything to this baby while they were still in the womb. What she found out is that working with the UNC here lab, how among the 7,000 metabolic signature that we detected in the newborn metabolome, we are seeing that there's actually over 600 of these metabolic features after false positive discovery, multiple testing correction. We are seeing that there's significant associated with at least one PFAS and at least one of the gestational age at birth outcomes. And more excitingly, compared to chemical standard, we're able to convert actually 56 overlapping metabolites shown here that are grouped into 7 major categories. Again, amino acids, proteins, bile acid become the top metabolism. And what's more excitingly is that compared to the maternal results of recording that we find out 10 metabolites in the maternal metabolome. And actually 8 out of these 10 metabolites also show up in the baby. And in addition, we found out that there are 43 other novel chemicals that in the newborn that are associated with both PFAS exposure and adverse birth outcomes. We also identified 14 unique pathways, and many of these pathways highlighted in red are again the same one that we previously identified in the mothers. And taking all these findings together, we propose this as potential molecular network that can kind of like connecting PFAS exposure and fetal growth restriction, which is centered around disruption in tissue neogenesis, neuroendocrine function, and redox homeostasis. Building on these exciting findings, we spent our most recent 2 years conducting additional analysis, trying to systematically unravel the mechanisms linking PFAS to reduce fetal growth through 3 different directions. One of them is mixture analysis. So 2 years ago, I started teaming up with Dr. Stephanie Ike, and we conducted one of the very first environmental mixture metabolomics investigation, where we use quantile G computation, QG-comp, to assess the joint effects of PFAS mixture on maternal metabolome. And what we found out is that when we consider the joint effect of PFAS exposure on the metabolome, we actually able to find out stronger and more disruption in the lipid and inflammatory bile acid related pathway that were perturbed when we look at the individual PFAS levels, which indicated that when we consider really the totality or the total effect of environmental chemicals, they really don't add along. We need to consider all these joint effects together. Another potential direction that we are pursuing right now in trying to understanding more from the causal inference pathways, whether or not some of these intermediators or this overlapping metabolite can serve as a mediator, a causal mediators of this PFAS and preterm birth. And this work is led by my other PhD student who just graduated, Dr. Susan Hoffman. She applied a parallel approach, not only just using the in-the-middle approach, but she also conducted the high-dimensional mediation analysis in parallel, trying to see if we can identify any novel mediators that seem to link PFAS exposure and reduce fetal growth. And what she found is that she identified 6 key metabolites that consistently showing up in both in the middle approach and the HEMA approach, with this particular fatty acids, omega-6 fatty acids, which are linked with higher PFAS concentration and increased risk of delivering a baby that's early term. And this is a potential positive indirect effect, which also indicated us that sometimes when we think about PFAS, we may also consider like nutrition and the role of nutrition during pregnancy in modifying this association. Another top mediator that she found out is the stress-related cortisol and cholesterol, which are both linked to preterm birth and mediating the PFAS early labor relationship. So maybe a little maternal depression could also play a role in this area. And now move beyond metabolomics, because this is just a single omics layer. In order to have a more comprehensive view of what's really going on in the body, I think we should look at other omics layer. So this is another newer study that we previously conducted. So instead of looking at the metabolomics layer, we also investigate maternal epigenomics by characterizing the whole genome methylation patterns. So focusing on the DNA methylation data from the maternal PBMC samples, my PhD student Yoran Tan compared more than 800,000 CpG sites and found out many of the significant association after the rigorous morpholomic corrections. And we found out that this single PFAS congeners seem to be linked to CpG sites that match to genes that are involved in cytoskeletal organization in cell growth pathways and in the lipid metabolism. And again, consistent with our result observed in the metabolomics layer, when we use QG-COM, looking at a mixture effect, we find out that many of these CpG sites, we actually discover more CpG sites linked to PFAS mixture and suggest that their involvement in regulating the genes that are critical for growth, for metabolism, and for cellular function. And later, she also ran a gene ontology analysis where she found out that this overlapping pathway are tied to fetal growth and placenta function, including the IGF binding, the CM signaling, and the steroid hormone metabolism. And recording about the steroid hormone metabolism, we also find out that particular pathway in the metabolomics layer. And this time, looking at the epidemiologic layer, we are seeing some sort of consistency across. And finally, some of the most recent interesting results, we find out that infant sex might actually shape or modify these epigenetic responses to PFAS, which indicated that in this particular crop, the take-home message is we find out that the baby boys seem to be more vulnerable or have more drastic association between PFAS and fetal growth compared to the girls. And we are still looking at this molecular mechanism more closely. Another omics layer in the pitch study that we focus on is lipidomics, because we are seeing so many fatty acids and bile acid metabolism perturbation previously. So this work was led by Dr. Caitlin Tybalt. Again, she identified numerous lipids in strong association with both PFAS and gestational age at birth outcomes. Specifically, again, we are seeing the most of the strong association when you consider the PFAS as a mixture. And we identified 11 lipid metabolites as a potential intermediates linking PFAS exposure and small gestational age. Most of them are actually related to phosphatidylalkanoids or saturated glycerates. And several of these contain bioactive fatty acids with strong association with PFNA, PFOS, and PFOA. This figure shows you that there are 12 overlapping lipid pathways with the key and top metabolic lipid pathways, again, in association with long-term depression, insulin resistance, and glycosophospholipid metabolism. And some of these are also picked up previously in the metabolomics layers. And finally, these enriched lipids seem to be highly correlated and interconnected with each other, which suggests the role of this lipidomics perturbation in inflammatory and oxidative stress pathway upon exposure to PFAS, which in turn increase the risk of fetal growth. So you may ask me, will you look at genomics or microbiome? Well, the answer is that yes, we want to do that, but we need to get funding, right? So one of the New Year wish that I have is that hopefully we'll hear some good news soon about one of the R1 application. And instead of looking at single omics together, we are trying to combine all these multi-omics together in order to have a more comprehensive view. And we definitely want to try with Dave's Lucid, this really powerful tool to do this multi-omics integration. And not only were we able to assess the mechanistic link between PFAS and birth outcomes, my team has also successfully applied this omics technology characterizing other ubiquitous environmental toxicants, such as tobacco smoke, as shown here, led by Yoran. Again, she discovered some integrated metabolic networks centered around the urea cycle and the TCA cycle, kind of linking exposure to tobacco and adverse birth outcomes. Another exciting example is our publication on phthalates, which some of you might know is emerging chemicals, a group of pesticides. We are finding that, led by one of my previous master students who graduated 4 years ago, she found out that numerous metabolic features seem to be associated with urinary phthalates metabolite, and they seem to be associated with thyroxine metabolism. And more excitingly, when my PhD student, Dr. Susan Hoffman, tried to replicate the study. She also found out that thyroxine metabolism and tryptophan metabolism are mainly perturbed when she focused on the newborn metabolome. And more excitingly, she found out that the perturbation in this newborn metabolome also seemed to be do some adverse effects on the newborn's brain development when we look at the infant N score. And this study was also like widely reported from CNN, and we are trying to work on some future application, trying to see like the infant neural development, how they got impacted by these pesticides. And in addition to this, we're also looking at other emerging pollutants such as PBB, PCB, bisphenols, pesticide, heat exposure, etc. And another major group of pollutant that I'm really interested in is air pollution. So in the past decades, many great exposure science and epidemiological study, including those led by our USC colleagues here, have revealed various adverse health effects upon exposure to both short-term and long-term air pollution exposure. And specifically, we also observed that higher level of air pollution during pregnancy will also link to higher risk of adverse birth outcomes, including preterm birth, which is defined a premature delivery before 37 weeks of pregnancy, and early term birth, which is a live birth between 37 and 38 weeks of pregnancy. And one thing that you will notice is that in the US, if you look at the yellow lines, these are the rate of preterm birth and early term birth for African American. And then the blue are the white people. And you will see that consistently they are at much higher risk of having this premature delivery compared to the general population. However, most of our current air pollution epidemiology study and molecular epidemiological study do not specifically focus on this population who are disproportionately exposed to higher level of environmental air pollution exposure such as PM2.5. So 4 years ago, I got my first R21 project from ISES to conduct the traffic exposure, maternal metabolome, and birth outcome. The TEMPO study, you will notice that this acronym is not as good as the PhD study, but I try my best. So here again, we use metabolomics, trying to understanding the impact of prenatal exposure to traffic-related air pollution on birth outcomes. Here shows you that for this particular R21 study, we focus on the 330 participants within the ATLA cohort. These are some of the inclusion criteria. And then for the exposure assessment, including like PM2.5, we use a fusion model developed by my colleague, Dr. Ted Russell at Georgia Tech, where we use RLI and CMAP model. We zoom into the spatial resolution on 250 meters, and then for the temporal resolution, we have focused on the daily concentration. We consider different exposure time window, trying to understanding both the short-term, intermediate, and long-term effects of air pollution, basically including 1 year prior to conception, average through the first trimester, 1 month before the blood draw, and 1 week before the blood draw. And shows you some of this slide shows you some of the general correlation, nothing surprising among all the exposure time window and distribution of the 1 year exposure. As you can see here, for those participants live very closer to the highway, we are seeing that they are exposed to a high level of traffic-related PM2.5 during their pregnancy, which is not surprising. Now, we want to see like whether or not we can understanding the underlying mechanism by using metabolomics. So this work is led by my first PhD graduate, Dr. Zhenjiang Li. He's now actually working with Dr. Leila Chassi at USC. So for the very first time, he adapted a very smart parallel strategy, again, using meet-in-the-middle approach and high-dimensional mediation analysis. Back at that time, we never used high-dimensional mediation analysis in metabolomics. This is a method that was first designed for epigenomics analysis. So using meet-in-the-middle approach, we actually identified 2 major pathways that seem to be perturbed a lot when the pregnant mothers were exposed to 1 year PM2.5 and both like short-term and long-term PM2.5 exposure. This includes the phenylalanine, thyroxine, tryptophan metabolism, as well as the protein digestion and absorption. They seem to be also associated with increased risk of having an early-term birth baby and a preterm birth baby. Then Zhenzhen were able to further confirm 10 metabolites with level 1 evidence that were found to be significantly associated with PM2.5 exposure after multiple comparison correction. I want to draw your attention to hydroxylysine. So what we found out is that when the 1-year exposure of PM2.5 increases, the intensity of hydroxylysine actually decreases. And when that decreases, then the birth weight or having a small birth weight of this baby also decreases, meaning that it's an increased risk of fetal growth restriction and an increased risk of early term birth. And hydroxylysine, in our knowledge, later we found out that it can be served as an anti-inflammation factors according to the existing literature, which means that here it could be served as a protective effect against this PM2.5 exposure. But then this protecting amino acid along with other got deprived when people got exposed to higher environmental chemical. And as for the high dimensional mediation analysis, we conduct this particular method using a R package called HIMA, HIMA, which again is originally designed for the mediation analysis on epigenomics data. And for the very first time, we are able to conduct this HIMA in a metabolomics study. And what Zheng Jiang found is that he found out that there's actually a metabolite that are significantly mediating the association between PM2.5 exposure and risk of early term and preterm birth. And very interestingly, 4 out of this A were also previously identified in the middle approach. And using both methods, we are able to confirm the identity of 12 overlapping pathways and metabolite. And here is the molecular network that we hypothesize here. But again, we need to really validate this finding in other independent cohort and using in vivo and in vitro models. And many of these pathway metabolites that we observed in the ATLA cohort were actually also reported in other independent cohort and independent air pollution metabolomic studies that I conducted over the past few years, including those that among healthy adults, among participants with or without asthma, among people who underwent assisted reproduction in panel study, crossover study. And not only do we see consistent and robust results for our own study, we are also very exciting to see that this pattern of consistent perturbation in the metabolite and pathway seem to be persist across different independent cohort, no matter which metabolomics lab that they're using, which study design that they're using. So we published this systematic review on air pollution metabolomics study, over 50 of them 2 years ago in EHP. And we are able to see some sort of like consistency, meaning that what we see is not just a chance finding or a fishing expedition. And hopefully we'll be able to further develop some of these metabolites as a sensitive internal biomarker capturing the internal exposure to air pollution in the future very soon. And building on all this work, last year I received an NIH R1 to go beyond metabolomics and to use other omics technology to systematically characterize the effects of prenatal exposure to traffic-related air pollution on maternal DNA methylation pattern, on longitudinal perturbation in the maternal metabolome, and also on the maternal gut and vaginal microbiome. And then our last aim is trying to bring everything together, which I'm really excited to work with this outstanding and super leading expertise faculty member here to really try to use like different omics, multi-omics integration together. And we have already got some exciting results. So using mixed omics package, one of the methods developed by a team at University of Melbourne and led by my PhD student, Dr. Susan Hoffman, we are able to see that there's more than 20 metabolites in the helix and C18 column, as well as more than 30 epigenetic CpG size hypo or hypermethylation patterns that seem to be associated with each other very strongly. It might be hard to see here, so the orange line is those participants' multi-omics DNA methylation and metabolomics profile among those participants who deliver a preterm birth baby. And then the blue line is those who deliver a full-term baby. So one thing you can see is that the expression pattern for these two omics layer differ substantially, and they have a very good prediction performances in distinguish between the mother who eventually deliver preterm birth compared to those who deliver full-term birth. And many of these CpG sites in the genes that got hypo or hypermethylated, and those metabolites seem to be enriched in inflammation and immune regulation. And then meanwhile, as part of Dr. Zhenjiang Li's previous dissertation, he also obtained some preliminary findings. This time, instead of using mixed omics, he used another omics integration methods called the MOFA, so the multi-omics factor analysis. And here, he identified several clusters of interconnected hypo or hypermethylated CpG sites and metabolite that are associated with both exposure to PM2.5 during 1 week or during first trimester, and then increased risk of preterm birth. One of the limitations for this previous study is that because we only started with 150 participants, so hopefully with the support from my current R1, we'll be able to increase our sample size to even 400 to 500. Then we can start teaming up with Dr. Conti's team later to use like Lucy and other omics integration methods to integrate all these different omics layers together. And beyond maternal and child health, I also want to showcase some of our work, how we apply omics technology in study other diseases, especially those chronic diseases ranging from asthma, cardiovascular diseases, Alzheimer's diseases, and lung cancer. So one of the exciting work that we recently completed is teaming up with Dr. Anka Hughes, also who is also a faculty member at Emory. We conducted high-resolution metabolomics on over 162 brain tissues collected from patients who unfortunately passed away from Alzheimer's diseases as part of the Emory Alzheimer's Disease Research Center. And here, focus on these brain tissues, we are able to identify 155 metabolic features associated with Alzheimer's disease pathology. And we also conducted pathway enrichment analysis, trying to understand the biological mechanism. So here shows you like over 36 metabolic pathway that pops up linked to Alzheimer's disease pathology. So when we look at the blood among those patients with Alzheimer's disease, we were able to only identify less than 10 or 12. But then when we look at tissues levels, we are able to actually pick up more granular changes in the molecular levels. And we are able to confirm 18 unique metabolites with level 1 gold standard evidences. And based on these findings, we propose a potential network describing how metabolites in our brain will be responded when people are having a more severe Alzheimer's pathology, which seem to be centered around the disruption of glucose and carbohydrate energy metabolism, as well as the dysregulation of the purine and pyrimidine metabolism, which is very important for brain health and for the DNA damage and repair and neural protection pathways. And this study will be published soon in the Alzheimer's and Dementia Journal. But not only that, we are able to also obtain through this really initial study, obtain enough evidence to convince National Institute on Aging in funding us to further support this work. So very fortunately, last year, our group obtained 2 major NIA awards. With one is an R1 study co-led by Dr. Anka Hughes and me. So this time, move beyond the brain metabolomics. What is cool about the brain bank is that before this patient pass away, we also have the matching blood samples. So we will be able to look at what happened in the blood and whether or not the pattern that we found out in the brain can be replicated there. So if we can detect something in the blood before these people pass away, we might be able to develop some sort of early detection markers in another independent cohort with like healthy cognitive functions in a prospective cohort. We also hypothesized that exposure to air pollution will lead to this increased risk of Alzheimer's disease by perturbing the brain and blood metabolome. And another more exciting major grant that we got last year is this $12 million UO1 grant, where I team up with Dr. Anka Hughes and Dr. Thomas Wingo. So this time, instead of just focusing on metabolomics, we go beyond other omics. We are going to conduct a proteomics, epigenomics, and genomics layer on more than 2,000 participants within these 3 different independent cohorts within the Emory Alzheimer's disease cohorts. We are trying to conduct both the modeling on the PM2.5, which is the most hypothesized environmental factors that can lead to increased risk of Alzheimer's disease, but we are also going to conduct an untargeted exposomics profiling to see if there's any other novel environmental chemicals that will go into our brain, into our blood, and in the spinal fluid that contribute to increased risk of Alzheimer's diseases. So for this study, we team up with leading exposure scientists such as Dr. Yang Liu, Dr. Ted Russell for the air pollution modeling. For the targeted and untargeted exposomics, we're teaming up with Dr. Dana Barr, Douglas Walker, and Dean Jones. And we also have a great team in multi-omics profiling and ADRD related dimensions. And for the remaining time of my talk, which I don't have much, I also want to showcase a little bit work on our exposomics and multi-omics investigation on the cancer outcomes. Similar to the great multi-ethnic cohort study, the MAX study at USC, we also have another cohort that is ongoing that I'm in close collaboration with American Cancer Society in their cancer prevention study. So cancer prevention study is a study that started in 1980s, and they follow more than 2 million people over time. And as part of the study, we have built up like a nutrition cohort that have more than 110 participants that contributed to DNA. It's a great biobank, and we also have more than 40,000 people with the pre-diagnosis blood samples when they are cancer-free. And then among these participants, more than 5,000 of them completed the metabolomics profiling. So really 5 years ago, starting teaming up with Dr. In Wang and Dr. Ryan Diver from ACS, that we conducted a pilot study looking at air pollution's effects on the human metabolome in a group of breast cancer patients and control. And what we find out is that on this more than 1,000 participants who contribute their pre-diagnosis blood sample, we are identifying 43 novel metabolites, and we also are able to replicate more than 20 metabolites that were previously observed in our systematic review in this initial analysis. And building on that analysis, 2 years ago, we started receiving support from American Cancer Society, the Emory Winship Cancer Institute, and the Robert Woodruff Foundation, where we started working also with Dr. Opa Patel, who's the PI of the CPS study. We are able to actually increase the sample size of the participant that available for metabolomic profiling. Nowadays, in this particular cohort, we have more than 9,000 participants that has metabolomics profile, and I see so much synergies between the group at USC and this particular cohort, because I know that many of you are also doing amazing things in different type of cancer. For this particular study that I'm leading, I'm specifically focusing on lung cancer. So this is study particularly led by 2 of my PhD students. One of them is Ziying Tang, who is now a NCI F99K00 fellow. The other is Sabrina Chow. 2 of them actually conducted single and multi-omics study to look at the molecular mechanisms underlying the impact of air pollution exposure on lung cancer. For Sabrina, she successfully identified a very novel metabolite shown here that seems to be associated with both exposure to this ambient criteria pollutant and increased risk of developing lung cancer among over 1,300 participants who provided pre-diagnosis blood samples. At the same time, Ziying received like NCI F99K00 award, where she further dive deeper in looking at the underlying mechanism, not just looking at metabolomics, but she was also going to look at the changes in the proteomics layer, as well as trying to combine all these things together. So last month, she just published a paper on proteomics that I'm going to tell you more about. But before I do that, I want to tell you a little bit more about the CPS nutrition cohort study. So all of these 40,000 participants, they contributed non-fasting blood samples between year of 1998 to 2001. And then we also, based on their residential address and history, we conducted the retrospective exposure assessment using the extent air pollution modeling, looking at PM2.5, PM10, nitrogen dioxide, ozone, SO2, and CO. And then we later look at the follow-up time for the lung cancer. And then we also have like match healthy controls. So this is showing you some of the basic population characteristic of both the cases and control. Here we work on a pair of 105 cases and control match on their age, sex, BMI, and so forth, and ethnicity. For the plasma protein profiling, we work with the O-Link platform. We are able to focus more on the inflammatory and oxidative stress related proteins as a priori proteins, and we are able to look at 484 distinct proteins. And what Ziyin published last month is showing that we are able to, when we link both the air pollution exposure and these proteins together, she was able to identify over 20 distinct proteins that are associated with individual air pollutions or mixture, and specifically IL-32, ADAM15, and IL-8 demonstrated consistent negative association with more than 4 of these ambient exposure indicator, and most of these proteins, especially are linked to immunity and signaling, something that's similar to our findings previously in the maternal and child health. And not only that, she also looked at the metabolite layer. So working with the metabolome company, we are able to complete this metabolomics profiling on these cases and control. There are more than 1,400 metabolite detected, and among them, almost 900 we know exactly what they are. Then we use mixed omics integration package again to identify clusters and components of highly correlated proteins and metabolites. Together, we are able to identify 3 major components. And then they are all shown here. So as you can see, it's not only just like between proteins and protein. I apologize, this is really hard to see, but the tail message is that we also identify some very interesting so the pink one are the proteins and green one are the metabolites, some very interesting, highly correlated cluster of proteins and metabolites together. And then we also identify and confirm more than 25 proteins within this highly cluster that are associated with both exposure to air pollution and different chemical signaling pathway, TNF, and cytokine receptor pathways. And for the metabolite, there are 8 metabolite that is significantly associated with both exposure and the sphingolipid metabolism, glycophospholipid metabolism, and taurine and hypotaurine metabolism. Then we try to understand all of this together. We find out that these actually proteins and metabolite are highly correlated with each other, enriched in the chemical signaling pathways, TNF signaling pathways, sphingolipid metabolism, glycophospholipid metabolism, and taurine and hypotaurine metabolism. And then when we conducted the joint pathway analysis, trying to bring them together, we again seeing this consistent pattern of disruption in all these signaling pathways. And looking in the future, right now, Dr. Ying Wang and I are working on a major cancer grant center application. Hopefully we hear some good news soon, where we will conduct more in-depth exposomics characterization, because we know that it's not just air pollution that can lead to increased risk of lung cancer. It could be radon exposure, could be due to smoking, due to metals, emerging chemicals like PFAS, or heat exposure might even play a role in this. So we're trying to do this like exposomics profiling as part of this lung cancer center. And then looking at how this individual behavior risk factor or nutrition is going to modify the association. And finally, dive deeper in the omics technology by conducting metabolomics, proteomics, and epigenomics together. So just to conclude my talk, however, you don't feel too overwhelmed with all these different results. But what I'm trying to tell you is that it's using this like cutting edge and emerging various omics technology, they do seem to give us some initial hints of like the molecular roadmap connecting different exposure to adverse health outcomes. As much as we are very excited about all this, we still don't know the exact mechanisms. That's why we need to team up with each other together to replicate all these findings in different cohorts. But more importantly, to use like hypothesis-driven protocols and to team up with benchwork scientists using in vivo, in vitro model to confirm the validity of these findings. And hopefully one day soon, we'll be able to develop some of them as a sensitive biomarker for future clinical research. And with that, I want to conclude my talk by really thanking all these different amazing funding agency, really none of this can be done with my trainees in my group, in the Emerge group. And I also want to thank every single study participants and our collaborators in making all this science possible. So with that, thank you very much.
Thank you. Thank you, Donghai. That was wonderful. So much work in so many different directions. Fantastic. So the floor, I mean, and you were on time, so we have some time for discussion. Question. Dave.
I have a question. I think I'm loud enough. You can hear me?
Yes.
Yeah, amazing talk, tour de force of just everything that you've done. Maybe just get your thoughts from what you've done. You know, what are you gaining from the integrated multi-omic analysis that you're not getting by just treating pairwise and other things?
Yeah.
Just because this is a big topic of, you know, what is the hypothesis of multi-omic integrated analysis?
Yeah. Dave, that's a $1 million question. I'm not sure whether I will handle this well, but I will try my best. So I do think it's very valid, and it will be actually making all this biological interpretation of this single omics finding more valid if we look at the multi-omics layer, because we all know this biological molecule, they don't act alone. So when I focus on metabolomics layer, sometimes I can see some of this metabolite will react in the way that I assume it will react, right? So if pollutant goes up, if a pro-inflammation marker, it goes up, so the disease risk goes up. But sometimes we don't. Even we try in different cohort in longitudinal analysis. Later, we find out that actually this molecule, they might have like its positive or negative feedback loop. And sometimes it's because the environmental exposure changes the epigenomics, changes the way that how this gene are modified and then how this protein are expressed. So I do think that's really important to look at other omics layer. And my current strategy is that we start with the single omics study findings first. That's how majority of the things I show you today. Then we try to integrate everything together to see if any of this makes sense. So far, I felt like one of the challenges in the multi-omics integration is that most of this integration are data-driven. So we look at correlation, we look at like the direction and magnitude of the association. But I do think that we need to team up more with the bench scientists to put in some a priori biological knowledge in trying to put all these things together. Right now, one of the challenges for myself and my group is that every time we can, because we are dealing with like tens of thousands of different omics signature, we always are able to find out there's some highly correlated genes and metabolite, but it is not a single PubMed paper publishing connecting these two together. So we really don't know like what is really going on there. So I felt like this field is still super promising, but we have so much things that we need to learn, and we need to learn all these things together.
Excellent, thank you.
Yeah, thank you.
Great, thank you. Howard.
Very similar question actually follows up naturally with that. So you mentioned that you need to work with basic scientists to try to see if they can come up with an in vitro or animal some other model that will be able to test some of the pathways that you think might be operating.
Yes.
Can you give an example of actually working with basic scientists on this? Because it's challenging. First of all, animal models often relate to animals that have different metabolism, different dose response curves. There's organoid models that could be used. Vita and her colleagues have been working with that in terms of some of the liver and PFAS stuff. It seems like it's a great need, but I haven't seen great examples of how that plays out.
That Howard, I totally agree with you. This is a challenge in the field right now, especially for my work in maternal and child health. There's just like no way for us to find out a in vivo model or in vitro model that can mimic really this human. So what I decided to do right now is to focus on more on the cancer area, because like for cancer cells, you can culture it. And one of the example that I have right now, I didn't have the chance to talk about it, is previously when we look at the metabolomics association with air pollution in this group of lung cancer patients compared to control in the CPS study, we actually find out that sphingomyelin and gamma glutamate was associated with both air pollution and increased lung cancer risk. What I'm working actually with some of the scientists, Dr. Aparna Kasanwala from Emory University Winship Cancer Society, her lab has amazing model, like those like cancer cell line model, not just like looking at lung cancer, but looking at specific lung cancer type and how this cancer will survive once we knock down this like metabolite and protein. So at least we will be able to see like whether or not this metabolite are in these like good markers of this like cancer survivorship. And if that appears to be having this like same dose response curve that we see between lung cancer and this metabolite, we are going to then further expose these cells to air pollution as the next step. But back to your question, in order to make sure that this kind of mimic what's really going on in the human body, we still have a long way to go. And I honestly don't have the answer yet, but we are keeping trying. And one of the things that I learned over the years is that when I started my faculty position, I always dream about having some of my lab being a dry lab, some of my lab being like the wet lab. But later, when I submit my first R1 saying that I'm going to conduct metabolomic study in human and in the animal study, the reviewer don't even want to bother to discuss this. So that's why maybe I'm good at data analysis, but I really need to team up with like basic scientists to work together. Raymond.
Great, Raymond?
Yeah. My voice is dead. Thank you. Donghai, thank you so much. I knew you were amazing, but like, man, this is unbelievable. And a star in our Exposure Science Society. Maybe another $X million question, you know, in all of our like exposomics, you know, and Nexus and all these efforts, I always find there's this like massive tension between the biomedical view and the environmental view, right? And all the work you're doing to me clearly sort of shows how you can have like gene therapies or medical interventions or pharmaceuticals or et cetera, addressing kind of all these pathways that fix and improve health.
Yeah.
And then, you know, like we're always talking about the totality of environmental exposures, at least in the external environment and the social and the physical and the chemical and, you know, blah, 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 blah. Like just looking at your plot where you had all the different air pollution mixtures and
Yeah.
had like 6 pollutants, right?
Mm.
It was clear to me like, oh, PM 2.5 gets like most of it, SO2 a lot of it, and then all the overlap to me is very fascinating.
Yeah.
In terms of trying to understand, well, what it is about that air pollution that's driving that, so we can go back to sources and control them and have
Yeah.
policy, right, that protects health. And that's just with 6 pollutants.
Yes.
You know, and so again, I don't know the answer, we're thinking hard about it, but how would you envision, let's say, the ideal, like, you have everything on the external, you have everything on the internal, and you want to do your perfect XWAS to discover, not hypothesis driven, to be honest, but to discover what it is that's driving these diseases. Sorry, I know there's no answer, but I love your thoughts. Thank you.
Well, I guess like that's a question for Dave, another biostatistics, bioinformatic pioneers in the field. Because right now, when you think about exposome, is tens of thousands of exposure. When we think about omics, is another like large, even more larger scale. And then when you think about all these health outcomes that were coming out from it, I felt like there's not like a perfect way to do that. Although we have many mixture analysis, some works better than the others, like we have QGCOM, BKMR, WQS, but we also need a better or like a smarter way of analyzing these things, in my opinion. Sometimes we tend to focus like single exposure, single association, but when you add more component in it, it's really hard to really tease this out, and drives me crazy. Sometimes like, why on earth when I put in QGCOM, some of the PFAS seem to be protective and the others seem to be matter when you do the single analysis, then it's totally different. So I also think that also makes me wonder if that is because when we analyze all this exposure simultaneously, one thing that we inevitably introduce is all these different types of measurement error. When you think about air pollution, even for those 6 different pollutants, we all know that they're coming from the ambient field. When you look at the individual exposure, people moving around, some people spend more time indoor than outdoor, then there's a type of measurement error that we introduce. Then when we think about PFAS, right? When you look at the blood level, and maybe there's like different things that we didn't control because we are working with non-fasting samples all the time. So there's maybe some also other errors. So I felt like when we put everything together, we also throw in a lot of uncertainties, a lot of errors there. That might also could be why now it's so challenging. I know that in an ideal world, we want to measure everything perfectly, but in reality, we can't. One thing that I do think it's important is to really like you said, like go through this like different consortium, working with our colleagues in Asia, in Europe. If we can do enough study and enough EWOS study across different places, at least we might be able to see some consistent signals that pop up. Then maybe we'll be able to zoom in that particular more critical factor to maybe design another prospective cohort study or really work with our bench scientists, going back to animal study.
Great. January.
So I have a question maybe from the bigger picture to like a more specific question. I do see that you have a lot of work starting from the metabolomics and then you add more of the omics.
Good.
For example, like what you found with PFAS exposures, a lot of are related with lipid fatty acids.
Yes.
Right?
Yeah.
And they are added.
Yes.
And then what do you see those metabolomics methods can help you understand the previous metabolomics findings?
Yeah.
Right, and then whether that will help to find maybe the potential targets that we can do with those.
Yeah. Thank you so much, Donghai. So I do think lipidomics is a very exciting layers. So for both metabolomics and lipidomics, we are actually using the same instrument, the high resolution mass spec coupled with liquid chromatography. One thing that we find out in the metabolomics layer previously, when we look at PFAS impact on maternal metabolome, we find a lot of lipids popping up, but that's the pathway level. We were not able to identify any lipid-related metabolites using that particular method because the metabolomics profiling is optimized for picking up as many metabolites as possible without specific focus on the lipids. So that's why when we work with Dr. Ding Jones and the lipidomics core at Emory again, we conduct this like untargeted lipidomics profiling, but this time we are using the same method, but we do not pay attention to animal acids or other things. We just focus on lipids. And within the lipids layer, we are able to find out what exactly is the lipid that is enriched in the previous like metabolism of lipid that are associated with PFAS. So we are able to actually narrow down to 11 lipids. The paper is still like under review right now, but I think some of them are the unsaturated fatty acids. Lipid. That is super interesting. I think that also pops out in some of your previous analysis as well. And we are also working on another work, which I think is quite interesting, that is also related to lipids, is think about what is the role of nutrition in shaping all this. Like we know that like we could get PFAS by just eating food, right? Different things. One thing that I noted, one of my students is working on right now is to look at the food frequency question now data that we collecting during pregnancy. And what we found out is that when pregnant mothers are in higher intake of some certain of PUFA, like omega-6 and omega-8, omega-3 or omega-6, we are finding that it kind of attenuated the adverse effects of air pollution or preterm birth. So that's like quite interesting. And right now I'm trying to go down to the metabolomics and lipidomics pathway to see whether this is true. Because like on the supplementation, we are seeing that even though there's two mothers both living closer to a highway, one have like a higher supplement, consumption of omega-3, they seem to be doing okay compared to the others. And also I felt like that's also bring us a little bit closer to the precision environmental health. We're thinking about like intervention strategy, right? Every time we conduct an air pollution analysis, we know that it's not okay to live closer to the highway because you got more exposed, but you cannot tell our participant to move away from it, right? So I think maybe nutrition could be one way to think about in terms of like the individual protection of their own selves. That's something that we are trying to brainstorm more.
Yeah. Our
Another question for you. So. One aspect of the exposome that is not, I think, getting sufficient attention is the time varying component of
Yes.
exposure profile, right, any individual. And the windows of vulnerability during life stage, where
Yes.
you might actually be most vulnerable to
Yes.
toxin effects. So among the omics that you mentioned, one that could play a role in trying to understand these windows of exposure is the epigenome.
Mhm.
And the epigenome, in some research studies, and our group has done some of this, has been able to show how the DNA methylation changed depending on whether it's early life exposure or midlife exposure, et cetera. And it also has the additional value, of course, of being something that controls gene expression.
Yeah.
And it might be somewhere in the pathway of disease. So in all the different cohorts that you're working with, are there ones where the exposures are really well characterized that can then give you a sense of the epigenetic signatures of those exposures that can then allow you to infuse more of a time, understanding of vulnerability?
Yeah, I first of all, I really am grateful for your great comments, and I totally agree with you. And then for the cohort that I'm working with, and I think that's why I'm so passionate in maternal and child health, even though compared to other area, the funding portfolio is relatively smaller. I think that early life exposure, looking at prenatal exposure is a perfect timing to look at all this, especially some of our previous work. We're showing that like this prenatal exposure during early and late pregnancy, how they get connected to the newborn. Because like before the baby was born, you're still picking up some of these biological changes. One of the pity that we don't have is this longitudinal critical time point measurement. So we are only able to capture like early and late pregnancy. So in an ideal world, in a perfect situation, I hope to maybe have multiple time points collected during the pregnancy and also looking at the changes in the DNA methylation level to see how this adds together. So I think maybe longitudinal sampling could be a way. And one thing that we do learn from another pioneer in the exposomics field is Dr. Michael Snyder from Stanford. His group, instead of looking at thousands of people, they only focus on less than 100 people, but they measure and conducted a monitoring for like 10 different time points. And then they're able to see the trajectory of the changes of this environmental chemicals and these changes in the biological layer, which I think is a clever design, especially if the funding is limited. One other thing that I didn't share here is that I do think that epigenomics layer is better in terms of its stability compared to metabolomics. For metabolomics, it's really a snapshot. Like if you take my blood in the morning, in the afternoon, you feel that maybe I'm so nervous right now that maybe my blood pressure will be goes up and there's something to do with metabolite. But in reality, in the field of maternal child health, we cannot control when the mother visited the clinic, for example, for the sampling. So one thing that I've been trying to do is to also time the diurnal patterns, like trying to understanding like what is the effect of diurnal pattern on the sampling on the metabolomics. So there's still an ongoing pilot project that we are doing. We actually sample some of our volunteer, our students, in the morning, in the evening, at noon, just to see how these like biomarkers changes across time, while keeping all the other condition constant. Because one thing that I think the reason why sometimes we do see a lot of variances in the metabolomics findings is really about the timing of the collections. And we do know that there's a circadian rhythm also going in. And to your point, maybe when we expand that to the entire pregnancy or like even a lifelong course, that the variances might be even longer. Roger.
Okay, I think we are right on time to grab lunch. We should say again a big thank you to Donghai for this wonderful. And thank you all for your attention. We have many coming seminars this year, so stay tuned.
Thank you.
Was amazing. It's great. So amazing.
I try to cut down my slides.
Yeah, I saw 1 minute done.
Yeah, because of the slide, I don't have.
So great, so we can go, try to have some break, then we'll.
Also good. Thank you.
Yes, it's a great.
Yeah, yeah, I started chatting a little bit about language. Yeah, this is some. Not good. Yeah. Yeah. I also work with guys.
Oh, it's not. Yeah, I need to go.
What do you got? Oh, okay, I see that. Details.
I just keep reading it all, whenever they make us check.
Oh, yeah. This is better enough, right?
Thank you so much.
Thank you.
Really interesting.
Thank you so much.
I feel like you broke everything down in like very simple parts. This is all kind of like.
Yeah, it's a lot of things.
Exactly, but it was very like digestible, so thank you.
Awesome. Thank you so much. Thanks so much for coming.
I'm just wondering.
Oh, yeah. No, you don't. No, it's like, woohoo. How on earth? Because I can go back to our conversation. Say we know, and then you're doing all this. This is insane. It's amazing.
But this is what really makes me excited.
Yeah.
What
Yeah,
the,
yeah, yeah.
administrators.
Oh,
That I do still want to be on the contrary exposures. Very bad thing.
You are. You're doing all of it.
Yes.
You do all of that. The question for you, because I know when you is you have it for that meeting.
Yeah.
Two people come and say,
Yeah.
You said, okay, because instead of meeting, I'll
Yeah.
take you out for a drink.
Yeah.
You sit and chill, it might be like.
Yeah, that would be great.
And then I take you to dinner.
Yeah.
No, it's not.
How does the applicant for you?
Somebody that yesterday, I'm gonna changing all the language. Apply money at our center, so
Yang was telling me
I'm
that
very alive.
he's also learning it. He's like.
Yeah. I was gonna be fine, but hopefully that means for us to actually. Fun, give it a break, unless there's other things you want to do.
Yeah, I mean, my purpose of saying, well, previously I say, I try to see how it should work is USC. I mean, I can't really have more with you guys, but many of you guys I haven't been to USC.
Yeah.
I also never been to this part of that. Previously, I was in tools to have.
Yeah.
It was a studio that's 15 years ago. That's my
Yeah.
first time being in the US, and then 5 years ago I was in Ventura.
Yeah,
GRC, like government research.
yeah. So I can video, we'll go back later.
Yeah.
Make sure you want to know, but yeah, I'll show you the resource and then you'll get a sense.
Thank you. You guys all can do that.
Yeah, you're